పాతదే వస్తుంది. పాతదే
ఎక్కడే
పండుగ
పాతది వస్తుంది, చివరికి పెట్టు. చివరికి పెట్టు.
Okay, log in. Are you able to hear me? Okay, it is. If it is visible, audible, everything, it okay message. MPL students, Super 60 boys, Super 60 girls, okay it is. Okay. We'll revise quickly the what we covered in yesterday, the portion with regard to the biotechnology. It is a very important topic. It is also a lengthy topic. It includes biotechnology as well as varieties applications. And in case of introductory part, René Descartes, the 17th century scientist, naturalist, philosopher, biologist of 17th century. From 17th century onwards, there has been the roots of what is the biotechnology. That is, all our human knowledge with regard to the natural science were directed for what it is the human comfort, that is the creature comforts, as well as what it is ethical values. And whole understanding of the natural phenomena became anthropocentric, that is human centered. And major utility of the biological world, the plants or animals, is used as what it is the food. Okay. And physics and chemistry contributed for what it is technology, industry, engineering. Again, all contributed for what it is the again human comforts and welfare. Okay. And offshoot of the modern biology, that is biotechnology, whose products, the process brought a great quality in case of human life. Okay, that is the first pair information. And with regard to what it is Herbert Boyer and Stanley Cohen. So Herbert Boyer Herbert Boyer, who belongs to Western Pennsylvania and who discovered a couple of restriction enzyme with useful properties. With the help of the restriction enzyme who cleaved the DNA, and cleavage in a particular fashion results in what it is the single stranded sticky ends at the end of what it is the double strand. And these clipped ends readily pasted as a precise exercise who called are calling it as what it is DNA splicing. What do you call it? DNA splicing. Okay. And Stanley Cohen, another scientist, Stanford scientist, who discovered ringlets of what it is the DNA, in addition to what it is the main genome, and who simply separated the plasmid and transferred to these bacterial cells, who developed the technology of isolation and transfer of the plasmid to the cell. And combining the process, that is the DNA splicing as well as what it is plasmid transfer technology, we can transfer the desired DNA fragments into the host cell to make the desirable protein as you desired. So there is a great breakthrough in case of the biotechnology by producing the desirable protein by transferring the desired DNA configuration. So breakthrough in case of biotechnology. Because once we transfer the desired DNA, it acts as a manufacturing plant for specific proteins. To that extent we completed. And in case of the first part of this biotechnology, the utilization of what it is live microbes, enzymes, its properties to produce useful products, and in process, what do you call it is biotechnology. We are using what it is live microbes and enzymes useful to produce products, and these microbes used in process. What do you call it is the biotechnology. Okay. And biotechnology earlier said it includes in vitro fertilization, that is test tube baby, DNA synthesis, DNA vaccines, and replacement of the defective gene, that is gene therapy, all includes in case of biotechnology. Curd making, bread making, wine making, also the part of the biotechnology. What this biotechnology? It is a classical biotechnology. Now biotechnology word used in a restricted sense. That means whatever the products, curd, bread, wine, we are producing in a large scale for humans. What do you call it is biotechnology. Curd making in home is also a biotechnology, but you cannot call it as a complete definition for the biotechnology. That is produced in large scale, it is biotechnology. And European Federation of the Biotechnology encompassing, that is combining traditional view as well as modern molecular emphasis to define what it is biotechnology. He said that definition: integration of natural science, cells, parts, molecular analogues to produce products and process, what do you call biotechnology. And the overall definition, it is exploitation of microbes at industrial level to generate useful products. What do you call it is biotechnology. To that extent, we completed in yesterday class. Okay. Now it is modern biotechnology. It is based on the two core branches, that is genetic engineering. The second, it is tissue culture, and it is called it as bioprocess. The modern word, it is bioprocess engineering. Okay, so bioprocess engineering, not genetic engineering. The genetic engineering, it is manipulation of genes, simply tailoring of the genes as we decide. That means altering chemistry of DNA or RNA. What is altering? Deleting or insertion of the gene. Altering chemistry of the DNA, that is deleting or insertion of gene. What do you call tailoring? As we decide. So altering the chemistry of the DNA means there is a change in genotype. There is a change in genotype. If you introduce it into the organism, certainly there is a change in what is the phenotype. So change in genotype, what do you call this? It is genetic engineering. So manipulation of the genes as we decide. What do you call? Genetic engineering. Alteration the chemistry of the DNA and RNA and introducing into organism as you change the genotype, then change in variety the phenotype. What do you call? Genetic engineering. And second one, it is bioprocess engineering. And the second, it is bioprocess engineering. Bioprocess engineering. Bioprocess engineering. That is whether prokaryotic cell or otherwise it is eukaryotic cell, it grows on a medium for what it is product. Whether prokaryotic cell or eukaryotic cell grows on a medium for giving what it is the product, any, whether it is a vaccine, protein or food products or any. And particular prokaryotic cell or eukaryotic cell should not be contaminated. So micro-free, that is uncontaminated environment. So we are creating an environment, ambience for sterility. ambience. Here ambience means, suppose ambience for look. You're keeping everything where it is in a proper way. But we require micro-free environment. It should free from other micro. So we are creating ambience for what it is sterility. So ambience for sterility is created by means of what it is chemically engineered process. Chemically engineered process. Suppose how can you clean this environment by spraying some chemical? Chemical engineered process, some fragrance it is giving. So placing beautiful flowers, it gives what it is certain beautiful environment. So here we require ambience for sterility. So you are using chemicals to eradicate certain other microbes. So chemically engineered process. So ambience for sterility is created. For proper growth of prokaryotic or eukaryotic cell to give what it is a product in high quantity. Okay. Whether it is proteins, enzymes, vaccines. Okay, it is. So ambience for sterility is created for particular growth of the prokaryotic cell or eukaryotic cell to produce products in large scale. Okay. What do you call? Bioprocess engineering. So ambience for sterility. How can you create ambience for sterility? It is ambience for sterility is created by chemical engineered process by spraying or using certain chemicals. Okay. And conceptual development in principles of genetic engineering. Conceptual development in the principles of RT genetic engineering. Okay, what is the concept behind in the principles of RT genetic engineering? As earlier said, in genetic engineering, manipulation of what it is the genes, tailoring of what it is the genes. And if you take the sexual reproduction, if you take the sexual reproduction, variations created. Sexual reproduction, variations created. And so in this new genetic setup it is going to be created. New genetic setup is going to be created in case of what it is genetic engineering. And asexual reproduction. In this, No variations which are created. It is genetic integrity is maintained. But it is genetic integrity, it is maintained. Okay, genetic integrity it is maintained in case of asexual reproduction. Variations which are created in case of sexual reproduction. Suppose it is foreign DNA. It is foreign DNA. Imagine it is a bacterial cell. Foreign DNA introduced into what it is bacterial cell. The bacterial cell multiply what change you notice here? There is no multiplication of what it is foreign DNA. It is foreign DNA. It is alien organism. If you introduce the foreign DNA into alien organism, it won't multiply because we introduce into cytoplasm of the cell. Okay, this is the first point. If you introduce foreign DNA into the cell, alien organism, it won't multiply. Bacterial cells divide, but it won't multiply. There is one point. If the foreign DNA it was introduced into what it is a cell, it is the foreign DNA introduced into the cell, it is foreign DNA, it is the host DNA. Okay. Now, it is the foreign DNA. So here, what is the difference here? It is foreign DNA, host DNA. So foreign DNA can able to multiply along with the what it is the host DNA. In earlier diagram, there is no replication of what it is the foreign DNA because the foreign DNA replicate along with host DNA. Foreign DNA replicate along with host DNA because foreign DNA becomes part of chromosome. It becomes the part of what it is the chromosome, main genome, and becomes the part of DNA in chromosome. So foreign DNA, now this DNA having what it is with ORI. Earlier you said in case of DNA replication, ORI, origin of replication. So foreign DNA can able to multiply along with host DNA if it integrates with host DNA, become the part of the chromosome, having the DNA having ORI. So foreign DNA linked to what is ORI of the host DNA, it can multiply, produce indefinite number of the foreign DNA. What do you call it? Gene cloning. Suppose this cell produce 100 cells. We are going to get how many copies of the foreign DNA? 100 copies of the foreign DNA. What do you call it? Is gene cloning. The second point here it is. So, unless it becomes the part of the chromosome, it won't multiply. As it becomes the part of the chromosome, it multiply. This chromosome having providing what it is? ORI. Linked to what it is? ORI. Okay. So, foreign DNA multiply and produce indefinite number of copies of DNA called what do you call? Gene cloning. Foreign DNA multiply and produce indefinite number of the copies of the DNA. What do you call? Gene cloning. Okay, it is a gene cloning definition. ORI, origin of replication. Okay. So, foreign DNA multiply and produce indefinite number of copies of the DNA. What do you call? Gene cloning. Because foreign DNA linked to ORI, origin of the host DNA. Not inserted in ORI, it is linked to what it is, ORI of what it is, host DNA. Suppose for example, a bacterial cell after division, many forms 400 cells. How many copies of foreign DNA? How many copies of the foreign DNA? Yes, it is 400 copies, 400 foreign DNA you are going to obtain. What do you call it is? What do you call the process? Pawan Sagar, Himalakshmi, Venkatesh, Krishida, Sheikh Muhammad, Rajiv, Vijay, Amshidhar. What do you call it as? Yes, gene cloning. What do you call it is? Gene cloning. So gene cloning should be done by linking the foreign DNA to varieties ori, not inserting in case of varieties ori. Link it to varieties ori. Suppose it is ori, not it is linked to OT, not it is inserted in case of variety ori. If it is inserted in ori is going to be deleted. So it is linked to varieties ori in order to produce indefinite number of varieties, the copies. Okay. What do you call it? The gene cloning. Okay. So foreign DNA linked to the ori of what it is the host DNA, multiply, produce indefinite number of copies of the DNA. So getting such indefinite number of copies of the DNA by linking to what it is ori of the foreign DNA, what do you call it is gene cloning. So we are going to see what is the gene cloning. We can leave for the next class. Okay.